Hello, my name is Kinjal Kankaria, and today is August 7th, 2025. Welcome to my presentation on hormonal control for implantation and early embryonic development. This is a fascinating and complex topic because it combines endocrinology, developmental biology, and clinical medicine in one seamless process. Early pregnancy is not just about egg meeting sperm. It is about creating a precise hormonal environment that supports a developing embryo inside the mother's body. Think of it as preparing a guest room. The space has to be made ready, the guest has to arrive on time, and both have to interact harmoniously. And if anything happens out of sync, the visit, which in this case is pregnancy, may not happen. So, let's first start with some definitions. So implantation is the process where a blastocyst, which is a tiny hollow ball of cells, attaches to and invades the lining of the uterus. This occurs roughly 6 to 10 days after fertilization. On the other hand, early embryonic development covers everything from fertilization to implantation, which includes cleavage, divisions, blastocyst formation, and the development of early supporting structures like the trophoblast and the beginnings of placenta. Clinically, it is crucial to understand this because around 50 to 60% of conceptions fail even before a woman knows that she's pregnant, and this is often because implantation doesn't succeed. This knowledge also guides infertility treatments, early pregnancy monitoring, and miscarriage prevention. So, by the end of today's lesson, I hope that you would be able to describe hormonal changes in the female reproductive system and that prepare the endometrium for implantation, explain the role of different hormones, such as HCG, progesterone, and estrogen for the maintenance of early pregnancy, be able to differentiate between the hormonal contribution from the mother and the embryo during early development, and be able to analyze how disruptions in these learned hormonal signaling can result in implantation failure or even a miscarriage. So, the uterus is only receptive to an embryo for a short period of time. That is called the window of receptivity, or the window of implantation. This timing is controlled by the hypothalamic pituitary ovarian axis, or also called as HPO axis. It begins in the hypothalamus, which releases the GnRH impulses, and these pulses control the anterior pituitary's release of FSH and LH, which are crucial in a woman's reproductive system. FSH drives follicular development in the ovary. As the follicles grow, they secrete estrogen, which stimulate the endometrial cells to proliferate. You can imagine estrogen as the construction crew. It builds a lush, thick lining with spiral arteries and glands. Then, a mid-cycle LH surge triggers ovulation. The ruptured follicle transforms into the corpus luteum, which secretes progesterone. And progesterone is like the interior designer. It changes the proliferate lining into a secretory one, which is filled with glycogen, proteins, lipids, and all of that good stuff to nourish the arriving fetus or embryo. And if progesterone levels are too low, or if they peak at the wrong time, the window of receptivity closes and implantation fails. This is why progesterone testing is very common in infertility workups. So, after fertilization happens in the fallopian tube, the embryo takes about 5 to 6 days to reach the uterus as a blastocyst. And implantation then unfolds in 3 phases. Apposition is the initial loose alignment between blastocyst and endometrial surface. Adhesion follows, involving molecular handshakes between the embryo's integrants and the endometrial extracellular matrix. Invasion is when trophoblast cells actively penetrate the uterine lining. This requires enzymes to remodel tissue, plus chemical signals like prostaglandins and cytokines that regulate immune tolerance and vascular changes. Progesterone orchestrates much of this, ensuring the uterus remains calm, meaning no contractions to expel the embryo, and that maternal immune system accepts the semi-foreign structure because of paternal DNA. In fact, implantation has often been compared to an organ transplant, except here, the maternal immune system is actively taught to welcome the newcomer. One of the embryo's first jobs after implantation is to signal its presence. It does this by producing human chorionic gonadotrophin, or better known as HCG, from the syncytiotrophoblastic cells, or trophoblast cells for short. HCG is structurally similar to LH, and it binds to the same receptors on the corpus luteum. This rescues the corpus luteum from degenerating, ensuring progesterone levels stay high throughout pregnancy. Without HCG, the corpus luteum would regress after about 2 weeks, progesterone would plummet, and menstruation would occur, ending pregnancy. Now, clinically, you might think that HCG sounds familiar, and this is because it is a very powerful tool seen in pregnancy tests. They detect HCG levels in urine, to confirm a pregnancy. Moreover, blood tests measure exact levels of HCG, which should roughly double every 48 to 72 hours in early pregnancy. Slow or plateauing HCG rises can signal ectopic pregnancy or miscarriage, while abnormally high levels might point to molar pregnancy or multiple gestation. Now let's talk about the hormonal shift. By around week 8 to 10, the placenta takes over hormone production from the corpus luteum. This is the luteal placental shift. The placenta produces progesterone to keep the uterus in a quiescent stage, preventing immune rejection and maintain the endometrial environment. This also produces estrogen, which stimulates uterine growth, increases the blood flow, and prepares the breasts for lactation. The placenta also engages in autocrine and paracrine signaling, which is like the local chemical communication that fine-tunes both maternal and fetal tissue function. Growth factors regulate placental blood vessel formation, ensuring the fetus gets enough oxygen and nutrients. If the shift is incomplete or delayed, progesterone supplementations can be life-saving for the pregnancy. Now let's talk about what happens when there is hormonal dysregulation. When these hormones are disrupted, the consequences are serious. A luteal phase defect means the corpus luteum is not producing enough progesterone. This could be because of poor follicular development or inadequate LH support. And the result? A lining that's out of sync with the embryo. So, results in poor implantation. In ectopic pregnancy, the embryo implants outside the uterus. HCG levels rise more slowly, which can help clinicians detect the problem before rupture, which is very severe and life-threatening. Abnormal HCG trends can indicate miscarriage, molar pregnancy, or even gestational trophoblastic disease. In assisted reproduction, also known as ART, progesterone supplementation is standard after embryo transfer that mimics the luteal phase to improve implantation rates. Some protocols also include low dose of HCG injections to stimulate the corpus luteum directly. To conclude, and to wrap up, let's revisit the sequence. The HBO axis prepares the uterus through estrogen and progesterone. Implantation occurs during the brief receptive window, aided by local factors and immune modulation. The embryo's HCG keeps the corpus luteum functioning until the placenta takes over. And the luteal placental shift ensures hormonal stability for the remainder of the pregnancy. This process is a delicate dance of timing and coordination. Any misstep can result in early pregnancy loss. Hopefully, this presentation solidified your understanding of our hormonal control of implantation and early embryonic development. And if you have any questions or anything that confuses you, I would highly suggest to create a detailed hormonal timeline from ovulation to week 10 of gestation, listing each hormone's source, target tissue, and effects. It will help solidify your understanding of how these hormones interact over time. Thank you, and have a great day.